So how do you put an elephant in a refrigerator?
Take the giraffe out.
Did you say open the refrigerator, put in the elephant, and close the refrigerator? Wrong. Open the refrigerator, take out the giraffe, then put in the elephant and close the door. See, I said it was designed to make you feel stupid. So this tests your ability to think through the repercussions of your previous actions. Or make you feel stupid. Just use a different refrigerator. So the Lion King is hosting an animal conference, and all the animals attend except what? Which animal does not attend?
All of them.
The elephant. He's in the refrigerator. You can put him in there. See what I mean about feeling stupid? So this tests your memory. Okay, there's a river you must cross, but it is used by crocodiles, and you do not have a boat. How do you manage it?
You ride across the crocodile.
That's what I thought. You jump into the river and swim across. Have you been listening? All the crocodiles
You
are
just
attending the animal meeting. I just said it's designed to make you a little stupid, didn't I?
I'm just going to assume they're all connected from now here on out.
Yeah.
This tests whether you learn quickly from your mistakes. It just makes you feel like you're not standing on solid ground and anything can change at any moment. Which is what you might feel like sometimes in this class. Okay? People have asked, last semester you may have asked your teachers or your friends, is File 431 more difficult than File 430? And the answer is, of course. Otherwise you won't start studying right now as you should be. The answer actually is, it depends, obviously, on what you like to study. There is still anatomy. There are still parts of things to learn the names of, okay, in File 431. But there is more of the mechanisms, kind of like how a muscle contracted or how an action potential is produced, that kind of concept. Where once you figure out the sequence of events and what has to happen before something else has to happen, it makes sense. If that's the way you like to study, then memorizing the list of names of things is totally boring and not fun and therefore difficult to do. So again, it depends on what you like to do. If you're a good memorizer, if you're like my oldest son who has almost a photographic memory, and he's lazy and he doesn't study because he can read one thing once before. He had a test this morning at 8 o'clock, and by 9 o'clock last night his fever was coming back and he hadn't studied. And I said, well, honey, he goes, Mom, it's a really easy class. It's one of those things where you can just kind of figure out the answer. So hopefully if he woke up in time this morning, he's passed his test. But it's something he doesn't have to think about. All right, if you have to me, it's more fun if it's like a puzzle. If I can figure something out, and then once I know it, it always makes sense, and it's not something I forget. All right. So again, whether it's easier or harder depends on which way your brain tends to take you. So once you figure that out about yourself, you know whether you have to really push yourself to study or adjust spend more time on the stuff you hate, kind of thing. So there are more mechanisms. To me, that makes it more fun. Even though my doctorate is in anatomy, I really prefer the combined course where anatomy and physiology are learned together than the anatomy-only way, which I teach at Sac State, which is surprising since I'm the anatomist. But the reason I chose to get my doctorate in anatomy instead of physiology, and I spent 2 years thinking about it, was because you can learn physiology without knowing much anatomy, but anatomy doesn't do you any good unless you also know the physiology that comes along with it. So that's why I pushed myself that direction, because I wanted both of them. All right? So, we are intense in this class, if you haven't figured that out already. But hopefully you will figure out that I'm pretty fair in the sense of I tell you just about everything that you need to know. All right. Most of it's in your book. Some of it may not be. I do emphasize the autonomic nervous system, so if you didn't cover that in Bio 430, you might want to set aside some extra study time to go back to that chapter and look at the autonomic nervous system. Okay, I won't go over it in lecture. I won't just say, all right, you already know this. But if it doesn't make sense, you may want to go back to Netter's and look at some of the diagrams for that. I also will tell you particular concepts that are difficult that will be on the test. Please listen when I say that. You would think by now you would have figured it out. But every semester when I say this will be on the test, I get about literally half the class missing more than half the points on that particular concept. So when I say this will be on the test, believe me, it will be on the test. Okay? You will see it because I ask that question or those questions every single semester. If I talk too fast, and I will, please don't feel uncomfortable about raising your hand and asking me to slow down or to say something again, because I just get in the role of it. I tend to do that. The way I use the PowerPoint and the whiteboard is the PowerPoint tends to be primarily illustrative. In other words, what I'm talking about is on the screen, but I write a lot of lecture notes on the whiteboard. For one thing, it helps slow me down. You'll notice in the lecture notes, did I do it backwards again? Just that one page. Nope, I did it backwards. If you're left-handed, you've got the lines on the other side. Lines are supposed to be on the right side. Maybe they are in the female one. Anyway, take notes. Highlight, put stars by the information that's there. These lecture notes, according to we had a former colleague that started our Science Skills Center who was quite critical and negative on teachers providing lecture notes. And I kind of half apologized to her and she laughed and she said, Marcy, those aren't lecture notes, it's in their textbook. So if I don't say it in class and it's not in the lecture notes, I don't test on it. Awesome. Angelina asked me, what did you go by? It's not Angelina. Angie? Me? Yeah.
Oh, yeah, Angelina.
Okay, I thought you had a different name. Anyway, if she could use a recorder, please do. I would love to see 20 recorders up here on the front desk. Okay? If you want to videotape or record a couple of semesters, some people did that and put them online. I'm going to have to talk to you how you get, I can't transfer more than 12 minutes of video from my phone onto YouTube, so I'll have to talk to some of you guys about how you do that first. So there's some old YouTube videos of this class from a year ago. For some people it helps to listen to the material. Again, you'll hear stuff that you don't remember me saying. Or you'll hear an emphasis or an explanation, an anecdote, an example of something you may not have heard the first time. It also helps you relax and listen. You don't feel like you have to be writing down everything I'm saying because it's captured on the recorder. Okay? And you can listen for the explanation. It also helps to preview the lecture notes ahead of time, because you can know if it's not in the lecture notes and it's in your textbook, I'm not going to go over it. So you cannot read that if you don't want to. It also helps you to know when you're previewing the material, okay, this is what she's going to talk about, this is what I should read tonight. So when you come to class, you will have heard those terms ahead of time. You can make marks about something that you might have had a question that you didn't understand, and that can encourage you to listen more closely or to ask a question if I don't explain it during lecture. And so use these lecture notes. The first time you come to class should be the first time you've heard or seen the material. You've either gone over the notes the night before or you've looked at the text. Okay? I just don't turn recorders on and off. I'll leave that for you guys to do. So we start off with the endocrine system. In last semester, I finished with the muscular system, but the textbook transitions from nervous to endocrine. Some do not. They put the endocrine at the very, very end. This organization, I'm a little bit different from most of the faculty in both semesters. This organization started out kind of on a mistake of mine about 10 years ago. I didn't get my order in for hearts for dissection, and so I had to change the order of lecture. And endocrine, the way I used to start off the second semester of the course, and then reproductive follows really nicely with that because of the endocrine control of reproduction. And so I've chosen endocrine first. We won't go over every single endocrine organ on Thursday when we look at organs Thursday and Friday. Some of the hormones for the digestive system we'll talk about with digestive system. For the heart and kidney we'll talk about with those organs. So we're going to look at mechanisms today, and we will cover the pituitary gland and the thyroid and parathyroid gland as far as endocrine organs in this unit. And then we'll move into reproductive system. So with each unit, there are hormones that are involved in regulating the activity of the organs in that system. And so we'll be pulling the endocrine system with us all the way through. All right, so the organs that are listed, the most common endocrine organs that people think of are pituitary, which used to have, and some people still call it the master gland. If you recall from Bio 430 and the nervous system, the hypothalamus actually controls the pituitary gland, and as we'll see on Thursday, actually is the site of synthesis for two hormones that the pituitary gland releases. And then most of you have heard the thyroid, the adrenal gland, certainly the reproductive organs are releasing hormones. But the heart makes a hormone. The kidneys make a couple of hormones, right? The stomach and small intestines make hormones. So there will be a few more organs than ones that we typically think of. Back in Bio 430, in the introduction to epithelial tissue, there were 3 locations that epithelial tissue could be found or identified in. Anybody remember what those 3 locations were? Not specifically organs, but more in general terms. Where do we find epithelial tissue?
On the surface of organs.
On the surface, so covering the body, and as a pleura or pericardium covering an organ. Okay, so covering the body or an organ. A second location, the most common locations were lining, that's the third. Lining, so lining the mouth, lining the respiratory tract, lining the urinary and reproductive tract. And then the third was glands, all right? And we had 2 different types of glands. We had exocrine and endocrine. Anybody remember how the 2 differed structurally? They're both epithelial tissues, so the cells are secretory for both of them. How do they differ structurally? One has a connection to the surface. So this could be a sweat gland or a sebaceous gland. If we're talking about the pancreas, enzymes that the pancreas synthesizes are sent to the small intestine via ducts. All right, so there's always this connection to the surface via duct. Okay, but the cells are still epithelial cells by characterization. Most of our endocrine glands actually started out appearing like this with the duct, but that connection to the surface disappears. Okay? So a duct is absent. But we still have epithelial cells. So the secretions then are distributed rather than via duct. The secretions are going to exit the cells and be taken up by capillaries. So because they're going into the blood, and blood goes back to the body and then to the rest of back to the heart and then to the rest of the body, these chemicals have a widespread distribution. Okay? And we call these chemicals hormones. So they have this wide distribution, something that's secreted by the thyroid gland going into the blood is going to go to the brain. It's going to go to your little toe. It's It's going to go back to the thyroid gland, but it may not have now. Thyroxine has quite a wide variety of tissues that it binds to. But maybe we're talking about luteinizing stimulating hormone, LH, follicle stimulating hormone, FSH. That doesn't have quite as wide a distribution. It goes to cells in the ovaries or the testes primarily. So what keeps luteinizing hormone from affecting smooth muscle cells in your stomach? Receptors, right? So receptors provide the specificity. Receptors are protein molecules on the surface of a cell. And so here we have hormones. The chemicals go in, and then there's a receptor either on the surface of the cell or inside the cell. And if there isn't a receptor, that chemical has no effect on that particular cell. So pica-internal cells have receptors for luteinizing hormone. Testosterone interstitial cells in the testes have receptors for luteinizing hormone. Smooth muscle cells in the stomach do not. So even though the chemical is there, it's not going to change the behavior of that cell. So how do we increase the effect? How do we determine? So the receptor, it has to have a receptor, but if we want a cell to be more sensitive, let's say that we want the cell to change its behavior when it's just a little bit amount of that hormone is present, how do we do that? So here's the cell. Here's the receptor. And here's the hormone. So if we want that cell to be more sensitive and to be affected, have a greater degree of change, without dumping a lot of more hormone into the system, what can that cell be triggered to do?
Add more receptors.
Add more receptors. All right? Or if we want it to be less sensitive, then we already have a lot of hormone in the system, but we don't want it to be as sensitive, we could get the cell to undergo endocytosis and internalize receptors. Okay. So the cell itself can be somewhat in control about how it responds to this. This is called upregulation and downregulation. So upregulation means more receptors are added to the cell surface. And downregulation means receptors are internalized. Now, as we'll see in a few minutes, something has to happen. Just because a hormone binds to a receptor doesn't mean that, boom, the cell changes. So we have to cause enzymes to be activated. Or in the case of testosterone or estrogen, where the receptor is actually already in the cell, we have to activate DNA and transcribe it and translate the messenger RNA. So this is going to trigger a whole series of complicated internal cell changes. Now, some of you finished up the semester with nervous system. We did it a few weeks before the end of the semester. So chemicals having an effect on a cell to which they bind to via receptor is what we talked about with neurotransmitters, right? And so these do act in a very similar way by binding to receptors and even inducing activities and changes in G proteins. That's what some of our neurotransmitters did, like norepinephrine. What's some differences, some similarities then, and may I erase the board? The similarities and key differences between the endocrine system and the nervous system. So if you put the nervous system up first, since that's what we covered first. Okay, what's something that's similar?
They require receptors.
Okay, so they both have chemicals. Binding to receptors. All right.
They trigger the next cell to do something else.
Okay. Receptor, chemical receptor binding. Induces a change in the cell's behavior. Is it always the same change?
No.
No. Acetylcholine can cause heart rate to decrease, but it can cause digestive activity to increase. All right. So even though it's the same chemical, it depends on what's in the cell and the behavior of the receptor. The enzymes that are in the cell under the behavior of the receptor as to whether it excites or inhibits the activity of that cell. All right, what about how rapid the response is?
It's pretty fast in the nervous system. I'm guessing it's like
Okay,
slow.
so a difference here, the nervous system is rapid. We're talking milliseconds, all right? Whereas this is slow to forever. We're talking minutes to hours to days. What about recovery? In other words, if I decide to flex and extend my fingers, I can do that pretty rapidly, okay, or do this kind of thing. I can do that pretty rapidly. Can I get my heart rate to go up and slow down that fast? No. It has a nervous control, so we'll come back and look at that, okay? But remember, this is a rapid response time because we have a short distance. The synaptic cleft space between the neuron and the muscle cell, or the neuron and the preganglionic and the postganglionic neuron is pretty short. So this has a short diffusion distance. This is getting into the blood. So it has to go back to the heart typically and then get pumped to the organ that it's going to get to. So that can be a while. What about recovery time? So stopping the action. This has a rapid recovery time because we can, again, back to acetylcholine. Our postganglionic neurons have an enzyme. Our muscle cells have an enzyme that breaks down acetylcholine. If you want to maintain and sustain a contraction, you have to keep releasing that neurotransmitter. What's the name of that enzyme? Breaks down. Enzymes typically end in ASE. Acetylcholinesterase. Okay, or if it's norepinephrine, it's taken back up into the preganglionic cell and broken down by MAO, monoamine oxidase. What happens with the endocrine system? We've dumped a bunch of chemicals in the blood. They're distributed out to the body, diffuse out of the capillaries, bind to the cell. These cells typically don't have enzymes that break them down, so the cells are going to not respond by downregulation, but that's going to take time. We still have a lot of hormone in our bloodstream, so this hormone processing requires breaking down by the liver, excretion by the kidneys, or conjugation binding to other proteins. And that all takes time. So we talk about a half-life. How long is it before half the number of hormones that are present in the blood are gone? And that is usually hours. So to illustrate, we have some chemicals that are called neurohormones. They're really a neurotransmitter, but they're released into the blood. And one of the best examples of that is the adrenal gland releasing primarily epinephrine. It's made by embryological nerve cells that are found in the adrenal medulla that came embryologically from nerve tissue but are kind of clustered and don't have dendrites and axons and stuff anymore. All right, so let's say you're driving down the freeway and you're in the carpool lane, but you forgot that it's the day that you were taking friends to school. You just got in the carpool lane automatically. And the lights come on behind you and the siren starts and you jump and go, oh my goodness, I'm in the carpool lane and it's 8 o'clock and I'm getting a ticket. What happens to your heart rate? Goes up, your hands start to sweat, and you pull over, and the lights weren't for you. CHP officer is going on down to some accident 5 miles down the road. So boom, the stimulus is gone. You're not getting a ticket. If it was the nervous system, right away your heart should be back to normal and your hands back to non-sweaty. Does that happen? No. That's because in addition to the sympathetic nerves releasing norepinephrine to your heart, to your SA node, and to the ventricular muscles, your adrenal gland has dumped epinephrine into the bloodstream. And it's still in your bloodstream. All right? So it takes a while for it to be cleared from your bloodstream. That's the half-life. That's going to take longer for that to happen. So the body can't turn on and turn off quite as quickly to hormones as it does to the nervous system. But they still work very closely together. In most textbooks, not all, but in most textbooks, the endocrine system is discussed right after the nervous system sequence as a way of regulating behavior, body behavior. Interestingly, as a side note here, one of the case studies when we get to the cardiac and blood and immune system is a true story of a woman from Southern California who was climbing. Her goal was to climb every major mountain in all the continents. She's done about, I don't remember, 3 or 4 of them. She came down with a virus and developed cardiac myopathy. Her heart weakened and enlarged without being able to functionally contract and move the blood. And she had to have a heart transplant in her late 20s, early 30s. She still had the goal of climbing mountains. And she continues to do so to this day. Now, when she starts to climb and needs more oxygen and is moving her joints, her adrenal glands are stimulated to release epinephrine, and her heart rate goes up. All right? Does it go up quite as rapidly as if she had her sympathetic nerve system connected to the heart? Because typically in heart transplants, they don't reattach the nerve. And she decides to stop and take a rest. Now, if you're in good condition, your heart rate returns to normal in 5 to 6 minutes. Hers isn't going to do that. All right, the heart rate will go up because the epinephrine is going into the bloodstream, but we don't have a hormone equivalent of acetylcholine. So the vagus nerve, which would normally slow her heart rate down, isn't releasing acetylcholine to the heart, and there's no hormone, there's no endocrine organ that does that. So she has to wait for the norepinephrine, the epinephrine, to be cleared from her bloodstream. And that can take 30 minutes to a couple hours while she's waiting for her heart rate to come down. Okay. So there are some benefits to that, but she doesn't have that recovery benefit. Now, in addition to the typical hormone that we talk about being secreted into the blood, we have 2 other terms, autocrine and paracrine chemicals. Okay, auto means same as. And autocrine, this may not be on your lecture notes, but I just wanted to cover it here. Autocrine means the same cell, let's say a cell in the stomach, releases a chemical that has a receptor to the same exact type of tissue cell nearby. It doesn't get into the blood.